Today we are going to discuss the molecules of the living systems. Now, life as we know it is an integrated result of myriads of interactions between different molecules of living systems, collectively called the biomolecules. To understand life, we need to consider certain key features of these biomolecules, namely the chemical properties and reactivities, their structure and stabilizing forces, and the different kinds and proportions, their mutual interactions, and their origin during the emergence of the first living cell. Now, in the present section, we discuss some of the chemical principles that dictate the properties of the biomolecules. For example, the covalent bonding of carbon with itself and with other elements, the importance of the three-dimensional structure, that is the stereochemistry of these carbon compounds as reflected in the biomolecular properties, and finally, the functional groups that occur in the common biomolecules. Hydrogen, oxygen, nitrogen and carbon, they are the most abundant elements in living organisms. Here you can see that they are the lightest elements in the periodic table that have the potential to form 1, 2, 3 and 4 bonds respectively. Now, in general, the lighter the elements, the stronger the covalent bonds that they form. Now, sulfur and phosphorus, they are the other two common elements found in the living systems. Sulfur usually participate in 2 covalent bonds. On the other hand, phosphorus usually participate in 5 covalent bonds. The chemistry of the living systems is centered on carbon, and carbon itself accounts for more than 50% of the dry weight of the cells. It can form single bonds with hydrogen atom, single and double bond with both oxygen and nitrogen atoms. The critical point is that carbon atoms has the ability to share electron pairs, that is form bond with each other to form very stable carbon-carbon single bonds, and this has profound biological significance. Each carbon atom can form single bond with 1, 2, 3 or 4 other carbon atoms. Moreover, 2 carbon atoms can also share 2 or 3 pairs of electrons to form double bond and triple bond. Double bond is quite common, but triple bond is relatively rare in biological system. Now, these covalently linked carbon atoms in biomolecules may form linear chains, branched chains, as well as cyclic structures. As a result, these carbon atoms can be covalently linked together to form the backbone of the different biomolecules, and they can have virtually unlimited variety of structures. Now, the 4 single covalent bonds that can be formed by carbon atoms are usually arranged in this tetrahedral geometry, where the angle between each bond is roughly 109.5 degree and the average bond distance is about 0.154 nanometer. Now, there is free rotation around each single bond like this, unless sufficiently large charged groups are attached to both carbon atoms so that the rotation around this bond is restricted either sterically or electrostatically. On the other hand, double bonds, they are first of all shorter, about 0.134 nanometer, and more importantly, they are rigid and permits negligible, if any, rotation about its axis. You can see the angle between the substituents with the carbon atom is roughly 120 degree. And these two doubly bonded carbon atoms and the substituent atoms A, B, X and Y, they all lie on the same rigid plane. Now, physicochemical properties of a biomolecule is greatly influenced by stereochemistry, the arrangement of the atoms and the groups in three-dimensional space. Now, compounds of carbon, including most biomolecules, usually exist as stereoisomers, that is, different molecules in which the order of bonding of the atoms and groups are the same, but the spatial arrangement of these atoms and groups are different. Now, molecular interactions between biomolecules, they are always stereospecific. That means the two biomolecules can fruitfully interact only when they have a specific relative stereochemistry. There are three common ways to illustrate the three-dimensional structure of simple molecules. Here it is shown using alanine, the amino acid, as an example, and each of these three representations, they designate the structure unambiguously. First of all, let us consider this flying wedge form. Okay, you can see in this structure, these lines, they represent bond on the plane of the screen. On the other hand, these solid wedges, you can see, they represent bonds in which the atoms at the wide end of the wedge projects out of the plane of the screen towards the viewer, and these dashed wedges, they represent bonds extending behind the plane of the screen. This is another representation called ball and stick model. Here you can see each stick represents a bond and each ball represents an atom. This model is a good way to show the relative bond lengths as well as the relative bond angles. This is the space filling model. Here each atom is shown with its relative van der Waals radius. Consequently, the entire molecular contour signifies the outer limits of the region from which atoms of other molecules are excluded. Now, molecular conformation is a term that refers to the spatial arrangement of these substituents that enjoy the freedom of bond rotation. Consequently, they may assume different positions in space without breaking any bond. Here, the example of ethane is shown. You can see that these groups can rotate around this bond and they can form a different structure here. So, this is one conformation, this is another conformation, and this arose just by rotation of these groups around the carbon-carbon bond. Consequently, a large number of different interconvertible conformations of these simple hydrocarbons like ethane are possible depending on the degree of rotation around the carbon-carbon bond. Now, it is not possible to isolate any of these conformational isomers or conformers because the energy difference between them is small enough to allow rapid interconversion of the different forms, almost 1,000,000 times per second. But in biomolecules, substituent functional groups that are either sufficiently large or sufficiently electrically charged may exist so that they impart restriction to the freedom of rotation around this carbon-carbon bond. This limits the number of relatively stable conformations in such biomolecules. Now, configuration is another term that denotes fixed arrangement of atoms in space in an organic molecule that depends on presence of either a double bond around which rotation is not allowed or chiral centers around which substituent atoms or groups are arranged in specific sequence. Now, the identifying feature of configurational isomers is that they cannot be interconverted without temporary breakage of one or more covalent bonds. Here, a type of configurational isomers are shown that depends on the presence of double bonds. These are called geometric isomers or cis-trans isomers, where the substituent groups are arranged in relation to the rigid double bond. Here, there are two molecules. One is maleic acid, another is fumaric acid. You can see in maleic acid, the two substituents, these carboxylic acid groups, they are arranged on the same side of the double bond. That is why it is called a cis isomer. On the other hand, this molecule, fumaric acid, here the two carboxylic groups are on the opposite side of the double bond and they are called the trans isomer. Now, maleic and fumaric acid, they cannot be interconverted without breaking of the covalent bond, and this requires the input of significant amount of energy. So, this is the basic difference between conformational isomers and configurational isomers. Now, maleic acid and fumaric acid, they are individually well defined compounds that can be separated from each other, and each has their own unique chemical properties. Now, a binding site, say on an enzyme that is complementary to one of these two compounds, will not be able to suitably bind the other one, okay, because the structural complementarity is not there with the other one, and this explains why these compounds have distinct biological role despite their similar chemistry. Another such striking example is found in vertebrate retina, where the initial event of light detection is the absorption of visible light by 11 cis retinal. The energy absorbed from the light, about 250 kilojoule per mole, converts 11 cis retinal to all trans retinal, triggering the electrical changes in the retinal cell that lead to nerve impulses. So, this is the structure of 11 cis retinal, and after absorbing the light, it is being converted to all trans retinal, and this triggers the electrical impulse. Now, a carbon with 4 different substituents is considered to be asymmetric or considered to be as a chiral center. Okay, the term chirality means handedness is derived from a Greek word chiros, that means hand. This concept is used in the context of those stereoisomers that are related structurally as the right hand to the left. Okay. This is due to the different spatial disposition of their substituents. Now, for a molecule with one chiral center, only two stereoisomers are possible. But for a molecule with two or more chiral centers, say n number of chiral centers, the number of stereoisomers possible are 2 to the power n. This is illustrated example of 2,3 di-substituted butane. Okay. Now, in this molecule, there are two asymmetric carbons. Consequently, 2 to the power n, that is 4 stereoisomers are possible. So, these 4 structures are shown here in these boxes. Some of the structures, they are mirror images of each other. This with this as well as this with this. They are enantiomers with each other or enantiomeric pairs. On the other hand, the other combinations, they are not mirror images of each other, so they will be called diastereomers. Like this structure with this and this structure, and this structure with this and this structure. They have no mirror image relationship. Now, Louis Pasteur observed that enantiomers, they share nearly identical chemical properties, but differ in a particular physical property called optical activity. Optical activity means interaction with plane-polarized light, resulting in rotation of the plane. So, in separate solutions, two enantiomers, they rotate the plane-polarized light in equal magnitude but opposite directions. Okay. If it is clockwise rotation, it will be designated as plus. If it is anti-clockwise rotation, it will be designated as minus. Consequently, equimolar solutions of these two enantiomers, they will show no optical rotation. They were called racemic mixture, a term coined by Pasteur. And molecules without any chiral centers, they do not rotate plane polarized light. Here it is shown diagrammatically using alanine as the example. There are two stereoisomers of alanine that are enantiomeric with each other, that is mirror images, where one you can see has rotated the plane polarized light in clockwise direction, so it is plus rotation. The other one in anti-clockwise direction, so it is minus rotation. So the direction is opposite, but the magnitude is same in both cases. Okay. Biomolecular interactions are stereospecific. That means the molecules can fit only when they have a certain relative stereochemistry. Therefore, a biomolecule must be named and represented by a structure so as to make the stereochemistry unambiguous. Several systems are there that are used for such molecular designation. First of all, the RS system. Now, in this RS system, each group attached to the chiral center is assigned with a priority. For example, the relative priorities of some common substituents are, like here, OCH2 greater than OH greater than NH2 greater than COOH greater than CHO greater than CH2H greater than CH3, and hydrogen has the lowest priority. Now, the chiral center is viewed with the group of lowest priority, that is 4, pointing away from the viewer, like shown here. Now, if the priority of the remaining three groups, that is 1, 2 and 3, decreases in a clockwise order like shown here, it will be called R configuration, where R comes from the Latin term rectus, means right. And if it is in the counterclockwise manner, then the configuration will be called S, from the Latin sinister, meaning left. This is another system called the D and L system, which is a little bit older system but still quite used, where the descriptors D and L relate the sense of chirality of any molecule to that of D and L glyceraldehyde. So these are the structures of D-glyceraldehyde and L-glyceraldehyde shown in both flying wedge and Fischer projection, where you can see that the OH group is on the right side for the D, and for L the OH group is on the left side. The CH2OH group is at the bottom in all cases, and the carbonyl group, that is aldehyde group in these cases, is at the top. The isomers of glyceraldehydes that rotate plane-polarized light to the right is called dextrorotatory or small d, that was labeled as capital D, while the isomer that rotates the plane-polarized light to the left, small l or levorotatory, was designated as capital L. Now, to name more complex carbohydrates or amino acids in this DL system, similar Fischer projections are drawn, where the CH2 group is put at the bottom and the group containing the carbonyl carbon, either it is carboxylic acid group or aldehyde group, is placed on the top, or the ketone group. Now, the D descriptor is used when the OH or NH2 at the penultimate carbon, like here. In this case, there is no penultimate carbon, only one chiral center. But in these molecules where there are more than one chiral centers, we are considering only the penultimate carbon. Now, if the OH or NH2 is on the right side, like in this case, or this case, or this case, we will call it a D isomer. On the other hand, if the OH or NH2 is on the left side, like this case or this case, we will call it L isomer. Now, the three-dimensional structure of biomolecules, which is a result of the combination of configuration and conformation, is the most crucial for their biological interactions. Okay? For example, a substrate or a reactant binding to the catalytic site of an enzyme for effective catalysis requires close structural complementarity of these two molecules. Similar structural fit is also required in many other cases, like binding of a hormone to its receptor on a cell surface, or even antigen antibody recognition.
Sir, can you show us an image showing the structural complementarity of biomolecules?
Yeah, I have one such slide. Like here you can see, this is an image of a macromolecule which is enjoying a complementary fit with a small molecule. Here the gray region represents the macromolecule, which is a segment of the TAR regulatory RNA region encoded by the HIV genome, and this colored molecule represents arginine, which is an analog of a residue of the protein that binds in this region. You can see that this small ligand has bound on the surface of this RNA at a pocket-like region where it is complementary to this region. And these two molecules are held with each other by a number of non-covalent interactions. Now, in living systems, chiral molecules are usually present in only one of the alternative chiral forms. For example, the amino acids, the building block of the proteins, they occur only as the L isomer. Okay. On the other hand, glucose, the monomeric subunit of starch, exists biologically in only one of its chiral form, that is the D form. Now, chiral compounds in living cells, they are produced in only one chiral form because the enzymes that catalyze their synthesis are also chiral molecules.
Sir, can the chiral compounds be obtained in its different forms artificially?
Actually, when a compound with a chiral carbon is chemically synthesized in the lab, the non-biological reactions, they usually produce all the possible chiral forms in an equimolar mixture. Okay, and the product obviously does not rotate plane polarized light because it is a racemic mixture. Now, the individual chiral forms in such mixture can be separated only by painstaking physical methods. For example, Louis Pasteur separated enantiomeric pairs using forceps. What basically we learned today? First of all, the importance of carbon in the context of biomolecules. Carbons are the atoms that form the backbones of the biomolecules, and the stereochemistry of the carbon compounds, the rotational freedom around the single bonds, and in some cases rotational rigidity, that determines the three-dimensional structure of biomolecules, and the structure itself reflects the property of the biomolecules. And we also knew that biomolecules can exist in different stereoisomeric form, but in the biological context, out of different possible stereoisomeric forms, only one form is selected, and that is biologically important form, because other biomolecules, they will recognize only that structure. And since life is a summation of the different interactions between biomolecules, so the importance of the three-dimensional structure of a particular stereoisomer is emphasized.